Check, check. All right, class, let's continue with our discussion on the adaptive immune system, stage 3. Let's focus on antigens. An antigen, recall, is anything that can bind to an antibody. If we use the term immunogen, which is not very commonly used, but is occasionally used, an immunogen is the same thing as an antigen. It's a synonym for the antigen. Anything that can elicit an immune response, that can bind to an antibody, as a general rule of thumb, is an immunogen. Now, there are some specific criteria that need to be met. It needs to have the correct shape, the correct size, the correct accessibility, and it should not be normally present in the body. Immunogens that trigger immune responses shouldn't be normally present, because if they were normally present, then we'd have lots of autoimmune reactions. And that does occasionally happen, but that's a disease state. That's not the norm. So let's talk about some example immunogens or example antigens. That would include an entire microbe, or you could talk about some substances that come from human cells, parts, or various molecules. So any cells or substance that can come from an animal. Now, when I say human, generally speaking, it's a human that's not you. So think of a donor transplant or tissue or transplant rejection issue. Every once in a while, when your tissues are acting as an immunogen, that's a problem. Hopefully that never happens to you. We could also have proteins. Proteins make excellent immunogens. They're pretty big, they're pretty easy to grab onto. Or any molecule that has a protein in it makes an excellent immunogen. Generally speaking, if we have just a repetitive polymer composed of a single type of monomer, that's not going to be a very good immunogen or antigen. So if we were to look at starch, for example, starch is made of long chains of glucose or chitin. Or those can act like an antigen occasionally. Talk to anybody who has an allergy. But generally speaking, repetitive polymers that have a single subunit in them are poor immunogens. If we look at these immunogens, aka antigens, they're going to fall into several broad categories. Proteins. Proteins are the big ones. Any kind of a protein makes an excellent immunogen. We can have a lipoprotein, a glycoprotein, a nucleoprotein. Notice the theme here, protein, protein, protein. So we can have a protein all by itself, or we can have a protein bound to lipid, a protein bound to a carb, or a protein bound to a nucleic acid. Hey, proteins make great immunogens. Or we can have a polysaccharide. Now, when we say a polysaccharide, I'm not referring to polysaccharides that are commonly present in animals or plants. I'm referring to polysaccharides that make up the bacterial capsule, aka peptidoglycan. And if you're unfortunate enough to have a gram-negative infection in your body, we could also have lipopolysaccharides. Lipopolysaccharides are a potent immunogen or antigen. Now, let's talk about molecular size and shape. How can this affect the immunogen products of an antigen? If we have something that's too small, it'll slip underneath the radar. It won't get caught in our security net of our immune system. It must be big enough to merit the attention of our immune system. So we need large molecules. And when I say large, I mean at least 100,000 Daltons or larger. And a Dalton is, if I recall correctly, it's been a while since I taught Gen Chem, but I believe a Dalton is the same as 1 atomic mass unit. You know, let's double check right now. I am not going to be able to continue talking until I answer that question. What is a Dalton? Dalton to AMU. Let's just say 1 Dalton to AMU. I have to change this to the old AMU. Ah, it is the same. Ah, they're synonyms. I knew it. Where was I? Yeah, that term Dalton just isn't used very often. All right, I digress. Back to topic here. So we need to have big molecules. Now, to give you some examples, or some frames of reference, how big is a glucose molecule? A glucose molecule is 180 AMU or 180 Dalton. And if we were to look at a water molecule is 18 Daltons. So 100,000 Daltons is pretty dang big in terms of the size of a molecule. But size alone isn't the most important thing. for being an antigen. We also need to have some variety to this molecule. If we have highly repetitive subunits, it's generally speaking not going to be immunogenic. Proteins are excellent immunogens. Let's go back a slide here just to emphasize the point here. We have protein, 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 protein. Dang, proteins make great immunogens. Why is that? Proteins are made of 20 different amino acids. So it's very, very rare to see a chain of the same amino acid, but we'll have varying combinations of these 20 different amino acids. So proteins are not highly repetitive, so to speak. There is lots of variation within a single protein molecule, which makes them an excellent immunogen. If we look at an insulin molecule, insulin has an AMU of 6,000 Daltons, or AMU. Seriously, nobody uses Daltons anymore. And it is immunogenic. Insulin is at the small end of the spectrum. So let's look at some good immunogens and some bad immunogens. We have, hey, proteins. We could look at bacterial or microbial cells. We could have foreign human or animal cells, plant molecules that shouldn't necessarily be in your body. What is one that is common right now this time of year? Ooh. It's yellow, it grows in the ditch, it has some photoreactive chemicals to it. Golden Alexandra, I want to say. Don't quote me on that one. Oh, no. Wild parsnip, there we go. Wild parsnip contains a molecule that causes severe blistering. We could also look at uric acid within leaves of 3, leave it be, leaves of 3, leave it be, poison ivy as well. So anyways, we have good immunogens. I have too many tangents going on in my head right now. We also could have some poor immunogens. And the emphasis for poor immunogens are repetitive monomers that are the same. So many of the same monomers make a bad immunogen. Let's talk about epitopes. Epitopes are very closely related to antibodies, or excuse me, antigens. The part of the antigen that specifically sticks to the antibody, or that the lymphocyte can respond to, is called the epitope. So if I go back a slide here, let's go ahead and erase some stuff. Here we go. Yep. Most of, what in tarnation? There we go. All right. Most of this protein is not going to get stuck to an antibody, but little portions of it, right here, this blue portion or the yellow portion, and these repetitive blue or yellow portions can stick to antibodies. So the part of the antigen that actually sticks to the antibody is called the epitope. So our epitopes are the primary signal to our immune system that our molecule is formed. That's specifically what on the antigen we are looking for to stick to the antibody. Sometimes we're going to have really tiny molecules that all by themselves do not elicit an immune response. Those are called haptens. Haptens are related to antigens and epitopes, and let's talk about how. If we have a hapten, or this tiny molecule that normally by itself doesn't elicit the immune response, but we connect it to a bigger molecule, the small molecule plus the bigger molecule combine their powers and can elicit an immune response. So by the combination of them does develop or contribute to immunogenicity. The carrier group, that bigger molecule, contributes its size and makes that smaller molecule become active or available to the immune system. So if we take a hapten and bind it to a carrier molecule, the hapten is actually going to act as an epitope. So let's draw some pictures here. Let's say we have our favorite protein. And this protein all by itself, yes, very lovely picture of a protein, doesn't trigger an immune response. And then we also have our favorite drug. And our favorite drug and our favorite protein by themselves cannot trigger an immune response. This drug is a hapten. But if I take our favorite drug and I attach it to our favorite protein, now I can have antibodies attached to it. So drugs are a good example of haptens. Metals, think of nickel or silver or other metals that initiate an immune response, or ordinarily innocuous household chemicals. So think of ammonia or bleach or aspirin. These haptens can develop antigenicity in the body by combining with the large molecules, such as serum proteins. These serum proteins that are present in our body, all by themselves are self-proteins, so our cells are trained to ignore them and not initiate an immune response. But if we combine a self-protein, which by itself doesn't trigger the immune response, with the hapten, all of a sudden now we are going to have an antigenic response. So here we can see some haptens all by themselves, and they're very poor immunogens. They don't trigger the immune response. But if we combine the mass of a carrier protein to the hapten, now that hapten can act like an epitope and serves as a binding site for an antibody. We also have alloantigens in our bodies. An alloantigen is a cell surface marker. These cell surface markers are going to occur within the same species, but not in all others. Now, I should emphasize that they occur in some members of the same species. The classic example of alloantigens are blood typing. So, for example, I am blood type A positive, along with 1/3 of the US population, but other people could be B negative, or O positive, or AB negative, or AB positive. These are all different antigens that are similar to each other, but are different from person to person to person within a population, or organism within a population or a species. These alloantigens that are responsible for our blood types are also responsible for the incompatibilities that occur during organ transfusions. Earlier, several presentations ago, we talked about the MHC molecule, the major histocompatibility complexes, and how those are responsible for organ donation failures or tissue graft failures. We also have superantigens. What's the difference between a regular antigen and a superantigen? Well, glad you asked. A superantigen is really, really good at triggering an immune response. So it's a very potent stimuli, particularly for the T helper cells. H-E-L-P-E-R, there we go, T helper cells. So these superantigens do a really great job of activating our T cells, particularly the T helper cells, and then that's going to initiate an overwhelming release of cytokines, because those T helper cells can then also activate the rest of the immune response and trigger the rest of the immune cascade. This is going to cause a high release of cytokines. So after our T helper cells are released, we're going to activate a lot of cytotoxic T cells, and then those cytotoxic T cells release gajillions of cytokines, and now we have cells dying all over our body very quickly. And if lots and lots of cells die in our body at the same time and release their contents to the extracellular environment, we can initiate or trigger toxic shock syndrome or other autoimmune diseases. An allergen, which is closely related to a superantigen, is anything that can provoke an allergic reaction. We discuss allergens in more detail in chapter 14, so you can go ahead and read ahead or wait and watch that video. Now, to process the antigen and present the antigen, we need to go through several stages. Not all cell types are capable of processing the antigens. Generally speaking, you need to work with an antigen-presenting cell, and these antigen-presenting cells are going to phagocytize the pathogen and then rip off the antigens from the pathogen and then hold the antigens up on its surface and say, hey, look what I found. These antigen-presenting cells include macrophages, B cells, and dendritic cells. Macrophages, B cells, and dendritic cells are all antigen-presenting cells. After the antigen or the pathogen's been phagocytized and processed, the antigens from the pathogen are put on the surface of the antigen-presenting cell, and they're bound specifically to an MHC receptor. So there's an MHC receptor that holds the antigen on the surface of the antigen-presenting cell, and then the antigen-presenting cell will migrate around the body and bump into a T lymphocyte and activate that T lymphocyte, particularly the T helper cell. So here we can see a generic antigen presenting cell. If I had to guess, I would call this a dendritic cell because it has lots of extensions of its cytoplasm. So this antigen presenting cell is going to phagocytize the microbe, digest it up, and then after digesting the microbe, it's going to take little bits and pieces of the microbe, bind them into class II MHCs, and then hold those using the class II MHC, hold the antigen on the surface of the antigen presenting cell. And then that antigen presenting cell will migrate over to a T helper cell, and using its class II MHC, that antigen presenting cell is going to expose the antigen to the T cell receptor. That T cell receptor is paired with CD4 and CD3. Recall, CD4 and CD3 are present on T helper cells. CD8, cluster differentiation 8, is present on cytotoxic T cells. And then our T helper cell is activated and we trigger the immune cascade. Now these antigens typically are going to need to be presented to a T helper cell first. And that T helper cell will then go and activate other T cells and other B cells. Every now and again, though, we'll have potent antigens that can trigger the immune cascade independent of the T cell. So it can go straight to a B cell, and this antigen all by itself, without being presented within that class II MHC, can initiate the B cell differentiation. So these T cell independent antigens can trigger B cells directly without any antigen presenting cells or T helper cells. It's worth emphasizing here, T helper cell is not an antigen presenting cell. Our antigen presenting cells are distinctly different from the T helper cells. Some examples of that include carbohydrates with many repeating and invariable determinant groups, particularly carbohydrates from bacteria. Think peptidoglycan or lipopolysaccharides. So, concept check. Please tell me which of the following is not an antigen presenting cell? A macrophage, dendritic cell, T helper cell, B cell, or all of the above are antigen presenting cells. You can rewind the video, pause it, check in your notes, or check your textbook, get me an answer. 1, 2, 3, 4, 5. It is not all of the above. One of these is not an antigen presenting cell. If we flip back several slides, 1, 2, 3. We have macrophages, B cells, and dendritic cells are antigen presenting cells. So the correct answer is C. The T helper cell is not an antigen presenting cell. If you have any questions, please feel free to post them to the discussion board or shoot me an email. Happy studies.